Okay, so we're going to go ahead and get started with our second part in the meiosis and sexual reproduction lecture. So remember in the first one we were talking about the cells that go through meiosis are called germ cells. It doesn't mean cells that make you sick. It's a collective group of cells in females, in the ovaries, and when we talk about in males, they're in the testes. These are the cells that will become eggs or sperm by the process of meiosis. So they don't start doing this until puberty, and then by the time we're hitting menopause, early 50s for females or late 50s into our 60s for males, we've run out of these cells. They're not making copies of themselves. They're simply starting with 46 chromosomes or 23 pairs and ripping that information apart to put it into a sperm or into an egg. And at the end, every sperm is going to be genetically unique and every egg is going to be genetically unique. This is why there's variation within populations that reproduce sexually. So, okay. So last lecture we were starting into meiosis 1. That's where we're at right now. So here's meiosis 1. And what we're looking at with meiosis 1 is cells sitting here in interphase. Okay, you got your chromosomes. In the case of us, we'd have 46 chromosomes sitting here in interphase. We move from interphase into prophase 1 of meiosis 1. So prophase 1, big key event. The chromosomes overlap. They cross over. They swap pieces of information. Whatever we want to call it, they're going to exchange segments, the official terms, that crossing over. That's the big event of prophase 1. I mean, the nuclear envelope dissolves, the centrioles appear, all those things happen in prophase. But the key event to remember is the exchanging of genetics between buddy chromosomes. Pair 1 exchanges with each other, pair 2 with each other, pair 12 with each other. We don't want to see pair 1 exchanging with pair 17. That's a mutation. That's bad. So we go from prophase 1 into metaphase 1. So in metaphase 1, the key is your pairs of chromosomes line up. The term tetrads refers to the pairs of chromosomes, but the pairs in metaphase 1, the pairs of chromosomes will line up. Okay, so pairs line up. Excuse me. And then as we go from metaphase 1 to anaphase 1, the pairs of chromosomes split apart. They're going to rip apart. Okay, so we've split apart the pairs of chromosomes in anaphase 1. After anaphase 1, we'll move into telophase 1, and we will have 2 cells, each cell containing 23 chromosomes. Okay, something I forgot to mention, I apologize. Remember, one of the events of anaphase is the beginning of cytokinesis. The cell is going to start to pinch inward, forming that contractile ring or implants the cell plate. That is happening here in anaphase 1, and eventually it will split the cell in half by the end of telophase 1. Okay, so at the end here, I'll show you, we should have 2 genetically different cells. That's the key. At the end of meiosis 1, after we've gone through prophase, metaphase, anaphase, telophase, there should be 2 cells side by side that have 23 chromosomes in them. Now, there's going to be a mix-in and a match-in and an exchange in of genetics. Each cell is going to be genetically different because of that crossing over that happened back in prophase 1. Alright, so we now go into meiosis 2. And the key here, at the end of meiosis 2, there will be 4 genetically different cells at the end of meiosis 2. That is the goal. Okay, so Meiosis 2. There we are. We have a cell over here and a cell over here. Each cell in the animal world contains 23 chromosomes. I shouldn't say animal world, in the human world. 23 chromosomes in each of these cells. Both cells go into prophase 2. They go into metaphase 2. Now the key here that makes this different, in metaphase 2, we simply have chromosomes lining up. So there's 23 here, and there would be 23 here. You're not going to see them paired up anymore. They've lost their buddy. Their buddy is in the other cell. So 23 chromosomes line up across the middle of the cell in metaphase 2. As we move into anaphase 2, the 23 chromosomes split, and you have chromatids going to opposite cells, or opposite sides of the cell. So chromatids now in anaphase 2. Cytokinesis is beginning, cells starting to pinch inward, a little bit of an indentation here. And then we move down into telophase 2, and we finish splitting the cell in half. Cut that in half. In each cell, there's 4 cells. You got 1, 2, 3, 4. Each cell will contain 23 chromatids in them. So each of these down here, 23 here, 23 here, 23, 23. So the end result is what we call 4 haploid daughter cells. Okay, so a haploid means half. Diploid is full. We often represent haploid with this little n designation. Each of these cells is genetically unique. Okay? Alright, so here's kind of the big overview, simplification. Meiosis, Meiosis 1, Meiosis 2, that process. And I highlight this. The resulting cells are not genetically identical to the parent cell. They're different. There's the mixing and the matching, the crossing over. That's what produces unique variation. Alright? So, in males of a species that undergoes meiosis, they will go through this process called spermatogenesis. Spermatogenesis, let me get that. Production of sperm in male. Okay, so males are going to begin producing sperm at puberty. When puberty starts varies from individual to individual. If we're talking about in humans, maybe 12, 13, somewhere in there, this is when the process begins in males. The location is the testes. The group of germ cells there start going through meiosis. Cell divides. It starts out as a diploid cell, full genetic material. We call it the primary spermatocyte. And then it goes through meiosis 1, splits into 2 spermatocytes. So you got one here, one here. Now they are considered haploid because they only have half the genetic material, 23 chromosomes. Back here, you had 46. So now you got 23 of these and 23 here. So here we had 46 of these. So, meiosis 1. Now both cells will go through meiosis 2, and the result will be spermatids. These little spermatid guys here. They're haploid as well. They're going to have 23 chromatids. So you got chromatids, 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 chromatids. And then they mature, they turn into the full mature sperm, they get their tail, and they're ready to do their thing. So that's spermatogenesis. If those sperm are not released, they get reabsorbed into the body, get broken down, basically recycled, nutrients released, broken down, and then a new wave of germ cells goes through this. So there's a continuous production of germ cells going through meiosis to make sperm. So with males, they are constantly producing sperm. Every day there's a new wave of these guys going through the process, making sure there's sperm. Sperm only lasts a handful of days before they're broken down and reabsorbed. So then there's a new wave of sperm cells constantly replacing the ones that are being broken down and reabsorbed. So there's should always be fertile, functional sperm in a male during the course of their reproductive life. Again, that starts at 12, 13 maybe, and will go until mid-late 50s, and then there's this trickling kind of gradual decline of production of viable sperm. At a certain point, though, there's no viable sperm anymore. Men are not producing sperm, and they're no longer fertile. They're still going to be alive for many, many years, but no sperm production. Okay, oogenesis is the production of an egg and three things called polar bodies within females. Okay, so the egg is genetically unique due to crossing over. Same thing as the sperm. Now the difference here, though, a little bit of a difference. When oogenesis is occurring, female starts out with the oocyte. It's diploid, 2N. So it's going to have 46 X's, chromosomes. That goes through meiosis 1, and it turns into secondary oocyte, or the haploid structure. But when it divides, the genetics divide evenly, 23 and 23, but the cellular structures, the Golgi bodies, the mitochondria, the cytoplasm, etc., that doesn't divide evenly. The smaller cell is known as a polar body. Now that polar body still contains 23 chromosomes, just like the secondary oocyte contains 23 chromosomes. Okay, so again, the genetics have divided evenly. It's just the other stuff in the cell that hasn't divided evenly. Now, that cell eventually can go through meiosis 2, and at the end, you have an egg. That egg should, if all has gone well, contain 23 chromatids. And then the polar bodies, 23 chromatids in those. There we go. Now sometimes they show 2 polar bodies. Most of the time there's going to be 3 polar bodies that are going to result. So we'll put that third. That's supposed to say polar body up here. And that third polar body is going to contain, there's our third polar body, going to contain 23 chromatids as well. So all the genetic material has been divided up. Started with 46 chromosomes, divided into 23 and 23 chromosomes, and then those get divided into chromatids, 23 chromatids in the egg, 23 in each of the polar bodies, and the egg sitting there. One egg ready to be fertilized. If the sperm gets to it, and remember the sperm contains 23 chromatids in its structure, you put those 23 together, plus those 23, and now you have this fusion, and you have a zygote that has 46 chromatids, and the first thing that zygote does once it's blended the genetics is it's going to go into the cell cycle, it's going to grow, it's going to replicate its DNA, and you're going to have 46 chromosomes, and it's going to begin going through the cell cycle for 70, 80, whatever years throughout the rest of the individual's life. But there, right there, your genetic code has been set. Half from mom, half from dad, creating the zygote, which is a diploid structure, 46 chromosomes in humans.